You can see it?
Yeah, we can.
Okay, perfect. Well, thank you so much for the lovely invitation. It's such an honor and really a privilege to present at this meeting. So I'll be speaking about mathematical and computational medicine, from data to models and back. And Civ was really influential in my career, I'd say. While we're all sharing anecdotes, when I was an undergraduate student, I went to his office and really asked for a research project, and he gave me a couple of suggestions and just was incredibly helpful and really helped me launch a career, I think. And I'll outline a couple of stories that have been a lot of fun that I've worked on in the last couple of years. So I have no disclosures. So I'll tell you two stories today. One is about computational modeling of tumor evolution. And the second is about non-coding RNA biology and modeling those networks. And I'll conclude with how my lab is now using this to really design RNA therapeutics for treatment of glioblastoma. So as we get started, let me introduce the clinical problem to you. So glioblastoma is an aggressive, it's an incurable brain tumor. It's actually the most common primary malignant brain tumor in adults, so affecting about 12,300 people each year in the United States, and about a tenth less, sorry, 1/10 of that in Canada. The incidence occurs around the prime of life, so between age 40 to 60 years. And the survival of this tumor is on average, or sorry, the median is at 15 months. The 5-year overall survival is 6.8%. So the standard of care hasn't changed in the last almost 20 years. And unfortunately, even though we know quite a lot about the driver genes and driver pathways that push these cells to become incredibly aggressive in the brain, we don't know, we don't really have great treatments, and targeted treatments have really failed. So I'll present to you kind of a unique strategy at the end of this talk that my lab is using to work and target this disease. So first, let's talk about modeling cancer evolution. So cancer is, of course, an evolutionary disease. And much like Darwin's finches, what happens is a cancer cell acquires a particular set of mutations that give it a selective advantage to survive in a very harsh microenvironment. And in the case of glioblastoma, this is in the brain. And the whole concept of studying cancer evolution has really come up in the last probably 10, 15, 20 years because of the genomic revolution. So we can actually monitor and track evolutionary changes in tumor cells as cancer develops, as we go from a pre-malignant to what's called a malignant lesion. And the idea is to really use an idea of evolution as a process and examine forecasting using evolution. So how can we design biomarkers that take advantage of this evolutionary process, much like how we know physical properties influence weather? Can we predict in which way a cancer is going to evolve using an understanding of evolution itself? And so what many people have done is they have kind of looked at one aspect of a tumor. So that's called within tumor heterogeneity. Or we've found out that, okay, inside a single tumor, the cells themselves are quite different. And perhaps a measure of that degree of difference is useful to tell us something about how aggressive the cancer will be or how resistant to treatment it will be. But can we improve on this? And so what I'm going to present to you is a computational study done a few years ago, where we actually basically wrote an algorithm to help us model this using cellular automata model, and then test different biomarkers that essentially take advantage of evolution. So first of all, can we simulate an evolving precancer? Can we measure evolution? And can we predict among these simulated tumors who does worse or who does develop cancer first? So this is a lot of words, but I'll simplify it. Essentially, this is a CA model. We have a number of parameters. A cell divides based on its fitness. A cell dies based on its fitness. And at every division point, we decide if a mutation occurs. If a mutation occurs, there's a certain probability with which the mutation is either conferring a fitness advantage or a disadvantage. And as the cells on the lattice divide, they don't move, but they can shift around other cells. We do allow for we do allow for, as the cells divide, to keep track of the genotype. And using this, we can essentially say, well, if a cell has acquired a certain genotype, perhaps it stains positive for a particular marker. And so we can kind of connect it back and say, if we have imperfect information, for instance, what a pathologist might actually have, how can we, reconcile that with the truth of this model? And so we looked at an outcome of interest wherein 5% of all the cells on the lattice or whatever cutoff became cancerous. So 5% of the cells acquired a certain number of mutations. And what we're able to do is ask a certain set of questions from this model. So how can we measure evolutionary activity using imperfect information? So just clinically visible changes. And we can ask questions about biopsy size. So if we restrict our area of information, how much more information do we get by changing the size or the number or the location of biopsies? And what features spatially are most concerning for a poor prognosis or a quicker time to having all the 5% of the cells become cancer in this lattice? So these are the questions we sought to answer. And the first thing we did was, well, we showed that traditional metrics work poorly. And for one example of this is something that histopathologists routinely do is they look at cells that are actively dividing. So when you get a sample of tissue, you can basically count the proportion of cells that are actively dividing. And if you use that as a marker of aggressiveness, many clinical studies have actually shown that it's not particularly useful. And we actually replicate the same here. So looking at the various quartiles, if you were to separate the various simulated tumors by the proportion of cells that are dividing, you actually cannot predict who develops cancer faster than the others. Diversity indices, so indices that quantify heterogeneity, actually do tend to work better. But the question is, can we do even better than these thresholds? And it turns out that what we have shown is that there's actually a class of indices that we've called proliferation indices. And what these do is these take into account the distance and fitness of the cells. So if we were to be able to tell, okay, a certain subset of cells carries a particularly aggressive mutation, if those cells are the ones dividing and they're close to kind of a cluster of cells that are rapidly dividing, these tumors tend to do worse because essentially they're creating a niche in which they can rapidly basically invade. And we're able to show that this works quite nicely. It really separates the the simulated tumors quite well. And actually we're able to predict outcome much better than anything else. So in summary of this part of the talk, I'll tell you that really traditional biomarkers in our simulated cancers showed a really poor correlation with endpoint. Index of positive proliferation, or IPP, which is a class of these markers, actually is a particularly strong predictor of prognosis. I didn't show you this data, but multiple biopsies, so we simulated this too, and if you take the average of many virtual biopsies, you actually get diminishing returns pretty quickly, so you probably just need one relatively small biopsy. And serial biopsies, so doing them at different time points and comparing the delta, was not particularly helpful in this very simplistic model. So really just using this very basic model, we're able to kind of ask these questions of evolutionary biomarkers. And so we're really looking at how do we take this and implement this with a computational pathology approach and try and bring this to the clinic. So the second part of what I'll tell you is also quite interesting, I'd say. It's about non-coding RNA networks. And in this study, really, we asked the question of can dynamics or nonlinear dynamics help us explain why there's so many species of non-coding RNA. And so it turns out that there are actually not just many, there are many species of RNA in the cell, and they exist in this soup of RNA molecules within the cytoplasm and the nucleus. And so everyone probably in the room knows about messenger RNA. These are the things that come from DNA that code for proteins. But there are also other species such as circular RNA, 3' untranslated regions, pseudogenes, long non-coding RNA, microRNA, and we don't know why all of these exist. There's no like real strong evolutionary reason why a cell should spend energy transcribing these molecules, and we've only been able to see them really because of how strong RNA sequencing has become, how deep we've been able to sequence within a single cell, exactly what exists there. And at first, everyone thought these were junk, but it turns out that they may have function. And I won't go into all of the biology of how they function, but I will show you a simple kind of mathematical model that we use to help understand some of this and pull out some interesting dynamics. So first of all, we have to realize that these microRNAs or non-coding RNA. So microRNA repress messenger RNA. That's their function. And they exist in a very smaller subset of feed-forward and feedback loops. And it turns out that these have very interesting behaviors. They can be used for noise buffering or bistability or oscillations or amplification of signal within a cell. And all of these have been experimentally observed. But this only answers the question for why do microRNA exist? Of course, we saw in the previous slide that there are like 4 or 5 other species. And so we really looked at something called a microRNA sponge. And this is something that is seen in nature. This is called a circular RNA. It has multiple binding sites for the same microRNA species, and it kind of floats through the cytoplasm and mops up these microRNA. And we asked the question, well, why does this exist? So we wrote a very simple kind of dynamical system describing this behavior with some time delays. And as you might expect, with time delays, you get very interesting oscillatory dynamics. And we, did the maths to show why or when these occur, what are the necessary and sufficient conditions. And it turns out that the presence of a sponge really can govern time-dependent oscillations, which is quite interesting and may actually be a good reason for these to exist, because there are many cases in developmental biology, and especially in cancer, where you would want time-dependent oscillations, for instance, the formation of vertebrae or as an organism develops, or even in cyclic hypoxia. And so all of these dynamics kind of can work together in this network, and we have shown that this is actually reasonable to occur. In the stochastic case, this gets even more interesting, but I don't have time to discuss that today. So we essentially did this modeling for the other species and have kind of shown that because of their degradation, production, and binding rates, they are used most likely for different things within the cells. And this really helps us start to understand this function of non-coding RNA and put together a greater theory of why we have these within cells and why they get dysregulated within cancer cells. I have 2 minutes left. So evolution can be modeled computationally with potentially clinically translatable biomarkers of disease. The dynamics of a new species of RNA can help us understand why these RNA species actually exist within the cell. And as I've shown you, I think non-coding RNA biology is an incredibly rich aspect of human disease. And I'll just spend the last minute talking about where we go from here. And this is my lab's direction. And really, at every scale of the disease process, we're looking at how we can integrate both modeling and data science to make a difference for patients. And each of these aspects is, I think very interesting. My lab is working actively. We're running one of the first clinical trials for wearable devices in patients with glioblastoma, where we're doing a lot of pathomics, as we mentioned, genomics. And I guess some of the people here are probably Siv's grandchildren. So just to show Siv the legacy he's essentially started. And our vision is really harnessing non-coding RNA as therapeutic agents, so giving them back into cells, particularly cancer cells, because we've kind of really studied their dynamics. And a successful therapeutic in this space has to really kill tumor cells, have clinical benefit, and we have to be able to measure this benefit and understand why resistance occurs. And so people in my lab are working on each aspect of this problem to rapidly translate this to the clinic. And so with that, thank you so much. Happy to take questions. I know this was a very quick talk, but happy to discuss further at any point. So just thank you all for your attention, and thank you for having me.
One quick question. I was told I'm a very bad chair, so I had to keep the time.
Okay.
Hi, Andrew. Thanks. A great talk. I have questions about the first part. For now, I had other questions, but I'm gonna skip them. So you assume, I guess the stuff that you didn't have time to talk, you assume some fitness landscape or fitness gains from your for the spatial tumor evolution problem.
Yes. Yes.
And so how do at the end of the day, what would that work out that your proliferating fraction of tumor cells would not be a good determinant for the treatment response or the stage of tumors? So what was your assumptions for proliferation or fitness gains?
Yeah, so it's all outlined in the paper. We studied a range of fitness gains. Essentially, each mutation confers a slight percent increase, and it's a constant percent increase per positive mutation and a slight percent decrease in fitness for a negative mutation. We then sample over all of those possibilities, and it's always proportional to the overall fitness, and it's a product of the positive and negative mutations for each cell.
And then you have also non-proliferative populations. You have also